and I'm going to try to share with you work that goes back essentially from what Bronwyn just said when I was a postdoc in England, but then has continued all up to the current day. And it's on a group of very unusual flies. You're not going to see one of these around here, but I will show you that there is a species in the same family that you can find here in Maryland in a moment. But let me tell you what my outline is. So I should also acknowledge the photographer that took these pictures. I sent him these specimens. He's a Swiss photographer who specializes in taking pictures of small things. And these are a couple heads of stalk-eyed flies. And what I'm going to talk about is I've got four sections of my talk. First, I'm going to talk about why do they have these crazy, almost always sexually dimorphic eye stalks, and then what do they have to do with speciation, reproductive isolation, which might happen before or after mating. Then, and you will see fairly early on that there's something that we call genomic conflict. There, we discovered a unusual genetic factor that interacts with sexual selection and influences reproductive isolation, particularly sterility in hybrids. And then I'm going to finally talk about where does that come from? What's the genomic origin? So there's going to be a lot of genetics in here. I'm going to try to make it accessible, I hope. But if it's not, throw a question in the chat or speak up and ask a question. I'm happy to slow down and try to answer any questions. So let me first start by saying that eye stalks in flies have evolved multiple times. And depending on how you count them, there's at least 8 different families of flies that have species that have broad heads. With Drosophila in Hawaii, you can find these Drosophila heteronura. They're kind of broad-headed males, less so in females. But then you can see these other species. This Richardia is from Costa Rica. Dopsisoma is from Peru. This platystomatid is from Australia. So they're found all over the world, but in all cases, it's the males that have the long eye stalks. And in these flies, the females actually have normal heads. And I should point out that if you look at the heads of these flies, they have antennae right in the front of their heads, just like a normal fly. And let me back up a second. If you look at this picture, the antennae of these flies are out here at the end of the stalks. So that's characteristic of the family Diopsidae. So all diopsids have eye stalks, both males and females, and they all have essentially split their heads so that their both eyes and antennae are out there at the end of those stalks. So many years ago, I was approached by a grad student at Yale named Rick Baker. That's a picture of him. And he was interested in developing a phylogeny for these flies. We had begun doing the same thing, so we collaborated. He collected flies in West Africa. I collected flies in East Africa and South Africa and Southeast Asia. And then we get some other flies from other people that sent them to us. And we were able to use DNA sequences to build a phylogeny for the group, for a subset of the group. There's about 300 described species and probably many more, but these are most of the main genera within the family. And you can see some of the variation in the heads of these flies. So this particular species here in the middle, that's found in Borneo. So the eye stalks are twice the length of the body in males. Then the species at the top is males and females are almost the same in terms of their eye stalks. So shorter eye stalks, and that's found in Africa. So all of these guys in this top group are African, then these are Southeast Asian, then there's some more African ones, and then the bottom group is actually found throughout the world. And this little fly in the bottom left, that was taken by a colleague at Georgetown and was caught in Rock Creek Park. So that is our local diopsid stalk-eyed fly. You're going to have to look close because they're only about 3.5, 4 millimeters long, and they're not really very abundant. You have to find them in the springtime or in the fall when they overwinter in crevices. You can find them aggregated, but they're not easy. We've actually caught them on the campus at College Park years ago, but I've looked and they're not easy to find. If anyone finds them, I'd be happy to take a few. Um, Interestingly, this fly next to it is actually a specimen in Baltic amber that's 50 million years old. So you can see they don't look a whole lot different. So the group goes back at least that long. Now let me explain these little boxes here. So this is plotting the eye span or the distance between the tips of the eyes on the y-axis and the length of the body from the tip of the head to the end of the wings on the x-axis. And the species where there is sexual dimorphism and where males differ from females, it's that line has a steeper slope. So in this case, big males have particularly long eye stalks, whereas small males actually are very much like females. So there's not much difference unless they're big. So that's a case of allometry, but it's sort of linear allometry. It's not curvilinear like many other systems. So you can see there are some species which are sexually monomorphic. There's no difference between males and females. You really have to look at genitalia to tell them apart. And what we plotted in sort of black and white on this phylogeny is how many times dimorphism has arisen, just yes or no. And you can see it's happened at least several times. But you can also, well, actually, before I go to the next slide, let me just show you how these flies come out of their pupae, because I think it's interesting to see. So this was taken by the BBC a number of years ago. So they basically swell up this part of the head, they pop out of their pupal cases, and they have normal heads for a while, but then they inflate the eye stalks. And they just go out in a matter of a few minutes. And you can see this, because they're transparent, you can see the nerve core or the nerve bundle. It's several thousand neurons that are actually running from the end of the eye stalk all the way into the brain, because they've split their eyes out there and the antennae, but they still have all the normal rest of the fly brain in the main part of their head. And you might also imagine that they can't really see very well right in front of them. They're blind, and they're blind right behind them, but they can see everything else around. So they actually have really good vision. If you find one in the forest, it will turn and look at you and it will respond quickly. They're actually pretty quick to try to escape from anybody trying to catch them. But they move their forelegs in front of them to sort of see what they're doing because they can't see. Anyway, so if you quantify that slope for the male and the female eye span to body length ratio, then you can see there's actually quantitative variation between these species. And what this slide Rick did for part of his dissertation is he showed that the change in the slope for males, if you look at two closely related species, that changes much faster than females. So the dimorphism that we see across these different species is being driven by changes in male eye span, not changes in female eyes. Females are kind of being dragged by the change in males, and they're dragged slowly. So you might wonder, why are they changing? And if you go back and read some of the early taxonomic literature, there's a lot of ideas like they're looking around obstacles with their eye stalks and doing various things, but I don't think that's likely to be the case. I think it's more likely it has to do with mating, and we've been able to look at this in detail in a couple species, which we can both study in the field, but also we've had in the lab, and we still have in the lab. So if you go out in the field, evening and you walk along stream banks in Southeast Asia and look underneath the banks where those roots are hanging down, you might come across this sort of aggregation with one male and a bunch of females. Um, and that's very typical. We caught these groups in Southeast Asia, and we typically find that the big males have a lot of females with them, and little males have very few, although sometimes the really little males are in with the big male because they look like females. You don't even know they're males until you catch them. We also know that if you bring them into the lab and you compete them, that males with larger eye stalks tend to win contests over food. And this is a picture. I didn't put a video of the fighting. I didn't have a really nice one like that eclosion picture, but they go through a very stereotyped set of interactions. They stretch their legs out beyond their eye stalks and they kind of match their eyes and their legs before they engage in more aggressive interactions. And they'll throw each other off of these little root hairs because the fighting occurs in the evening. In the morning, whatever male is there will attempt to mate with all those females. And we've seen males mate with up to 20 females in a half an hour. I mean, there's massive mating and then they all leave. Um, and we've also been able to show in the lab, create these little mating competition chambers where we put a little partition with hole punches that were big enough to allow females to go back and forth, but not the males, because the males' eye stalks are too big, so they would not get through. But the females could go back and forth, and these flies mate over and over again. Some flies don't do that, but these flies do. So we just count matings and have been able to show that the males with the longer eye stalks get more females coming to them. And there's a group in London who was doing the similar things after we did. They showed the same thing. And so there are definitely female preferences for longer eye stalks. And then finally, there's a genetic basis to this, and I'll show you more about that in a second. But it also depends on the rearing environment because the big fly is a fly that's had a lot of food when it was a larva. The small fly did not. So being a big with really big eye stalks is a combination of good larval environment plus having the right genetic composition. And I can say that because I took them into the lab, and one of the first things I did actually was started an experiment where I selected for longer eye stalks. And I did that by measuring 50 males, and I take the top 20% or the bottom 20%, use those to then mate with a random sample of 25 females. And those were the high lines. I had replicates or the low lines. And then I had control lines where I didn't select at all. I just put them together. And what I'm showing here is the difference between the average eye span distance of the eye stalks for the high lines minus the control lines. So it was the response for selecting up or the response for selecting down for these replicate lines for the first 20 generations. We continued this selection experiment for 70 generations, but I'm not going to show you all of those data because this makes the point. And notice that even though we did not select on females, but females still changed. So there's definitely a genetic component to the effect of the eye stalks in females in addition to the males. But the surprise came when my technician came to me and said, I can't find any males in one of the low lines. And so then we actually took 100 flies from each of these four lines and collected offspring and counted the males and females. And these figures show what we found. So that particularly in this second low line, 25% of the males produced all daughters. And in the other low line, a number of them produced all daughters, not quite as many. In the high lines, there wasn't much of a pattern for either way. It was not obviously different. And we thought, wow, what's going on? Well, a lot of other experiments that I'm not going to go through, but it turns out that the all daughter males have essentially defective sperm. Half of their sperm don't develop properly. And we're pretty confident those are the Y-bearing sperm. So flies, like humans, have XY chromosome composition in males, XX in females. So if you don't have Y-bearing sperm, you only produce X-bearing sperm, you're only going to produce daughters. And that's what these males do. They produce either 100% daughters or 90%. So very extreme. And you can count this. So these are, if you look in the testes, these are sperm bundles, 128 spermatids in every bundle. They should be half X, half Y, but half of these guys are look like they've been singed. It looks like hair that's been burnt. And pretty confident those are the Y-bearing sperm. Oops, sorry. And this was where I actually acknowledged my postdoc at that time, Philip Johns, who's now over in Singapore, but he was deep involved in this. And so we realized this is a sort of classic example of X chromosome meiotic drive, which I will just call X drive. So it is actually currently being used as a mechanism of pest management because you can engineer gene drives using basically the same idea. But this is a natural occurring. This is not something that's been engineered. And it's trying to illustrate in this little figure that you've got an X that's got an X a drive a element factor, something on it, and a Y-bearing sperm, and that X drive is producing something that's causing the Y sperm to not develop. And consequently, you only get X sperm. The female egg always has an X chromosome because it comes from a female. She's homozygous for X, and that produces largely daughters. That means that in contrast to a normal male that would only produce half daughters and half sons, this male is producing all daughters. That's going to increase in the population rapidly. And that element is going to increase in frequency. So this has been known for 100 years, and a lot of evolutionary biologists have studied this in other systems, never been found in these flies before. But one of the things that people realize that if nothing else happens, this should cause a population to go up in population size and then crash because you run out of males and there's nobody around to mate anymore. But there's also the possibility that maybe sexual selection might play some role here. And so I happen to be on sabbatical in Oregon, where this guy Russ Landy was, and he's done a lot of models of sexual selection. So we talked about this and came up with this idea that if there was something that a female that indicated males carried this aberrant X chromosome, the drive X chromosome, then a female that used that to decide who to mate with, in other words, to avoid mating with such a male, would actually produce more grandchildren because she would be able to produce sons. And that would lead to very rapid evolution of female preference. And sexual selection might even eliminate the drive, or depending on how good it is, how accurate it is, it might lead to some sort of balance. So that predicts that you would see the drive factor and the genes associated with the eye stalks somehow together on the same chromosome on the X and be in linkage. I mean, which means they're genetically connected. So again, Philip here, the postdoc, did massive experiment, lots and lots of flies, a lot of genotyping of flies. We went through 2 generations to be able to map, to identify where the factors that affect the eye stalks and the factors that affect drive, or basically the sperm, was what we used to identify the drive, and found that huge factors affect eye stalks are on the X chromosome, factor one, and the second is that there's not so normally if you like females have 2X chromosomes, you'll get recombination. The genes will interchange it before they segregate into the eggs for the next generation. We did not see evidence of any recombination between these two X chromosome types, and I'll come back to that in a while. Um, but then, roughly 10 years later, a group from London was out in Southeast Asia catching these flies and genotyping them the way we were, and it was able to show that you could just catch the flies in the wild, and they are clearly different. The drive carrying males have shorter eye stalks than the non-drive males. And this is a measure of how different they are from our lab reared flies. I mean, it is visibly different. So seems conceivable that females could be using this actually to make choices. So that motivated a postdoc. This is much more recent now. In the last paper came out a couple years ago. So Kim Paxold, who currently is actually a lecturer in biology, she teaches genetics, but she was a postdoc at this time, and she got funding to do an experimental evolution to test to see whether this drive is influenced by sexual selection. And she did it by, again, a massive experiment where she had 4 treatments, 3 replicates of each treatment. And the treatments differed by how mating was allowed. So the first treatment was 1 male, 1 female in a cage for 5 days. So there's no opportunity for any kind of multiple mating or females choosing or males fighting. Nothing like that can happen. It's just you get the male that we give you, and that's it. Second one is males were transferred between 5 females over 5 days, so females could mate with more than 1 male, which would allow for sperm competition, but not allow for males to fight because there's only 1 male in the cage, but every day the female got a new male. The next one is more or less natural, where you put a group of males and females together. They can do whatever they want. They can fight. They can choose. And the last one, and I should have acknowledged these first 3, we seeded the flies with animals that carried the drive X. 30% carried the drive, 70% did not. The last, this sort of control, is there's no drive. There's no drive X at all. So I'm only going to show you one result from this experiment. Took several years, a lot of work, a lot of help from students. So what this figure is showing, she did it for 11 generations. The generation time is about 2 months. So these are more like mice than normal flies, little flies. It's a lot of time and effort to do this. But this is the black line is showing the frequency of males in the lines where there was no drive. So it should be 50%. It was pretty close to 50% every generation. So that's good. The colored lines are these different mating treatments. So this red one is the paired single mating. And it goes down quickly and then it sort of stabilizes, but there are these red X's. So the red X's are actually, and there's 3 replicate lines, and these are the averages. The red X's mean one of those lines actually all the males were gone. So we had to actually breed extra males in order to have some males to keep that line alive. So the single mated lines continually basically went extinct, as you might expect, because drive got rid of all the males, and so there's no males left. If there's multiple mating and either the serial mating or the unlimited mating, you don't see that. We don't lose the lines. They continued fairly well. And if we estimate the frequency of drive, which we do in the prior generation genotyping, it ends up around 20 to 30% after 10 generations. The average single goes up and would have gone to 1 if we would have let it. And the wild population is roughly 25 to 30%. It's roughly what we got after 10 generations of this multiple mating, either multiple mating scenario. So we think this shows that sexual selection does kind of help balance the whole polymorphism for drive, and may explain to a large extent why you go out in the field and you catch these flies and they're still showing this. Because females mate multiple times, and that is enabling the non-drive to outcompete the drive, which is otherwise would have spread fast. So that's part 1. The other parts are going to be shorter. But sexual dimorphism, I think, in these flies is due to sexual selection, both because males fight with each other and big males win, and because females like males that are have longer eye stalks. Males that have this drive factor have shorter eye stalks, and there's a connection between drive um, and eye stalks because of reduced recombination. We think sexual selection is helping to maintain it. And now, what about speciation? So I'm going to shift and move on. Unless there's any questions, if people with me, I'm happy to take a pause if needed. All good. All right, let me push on. So this fairly early on, so we're going, this is the region, the Sunda Shelf, Southeast Asia, this is Thailand, this is Peninsula Malaysia, this is Sumatra, Java, Borneo. That's where these flies are found. So we've gone there, we've gone all over the place, catching flies where we can find them. We bring them back alive, and then at this point, we did some DNA sequencing and tried to figure out how they were related to each other, whether it was one big population or more than one. And we found at this moment that almost every place we went, if we sequenced it, it was fairly distinct from all the other sites. And this was a combination, if anyone's familiar with this sort of approach, we had a couple genes in the mitochondria, which are easy to do, and a couple nuclear genes, so it's not all mitochondria. So fairly robust, I would say, differences, even though if you go to the Natural History Museum downtown DC and go to the collections and look at their specimens, they will call all of these things the same name. So even though they're actually physically, they look quite different. There's some major differences, but they're all been given the same name. So there's clearly some cryptic species that have not yet been named. And what we did, grad student at this moment in time, Sarah Christensen, started trying to mate these different flies together and discovered that depending on how close they are on that phylogeny, which is a measure of genetic distance, they are more or less likely to produce offspring. And if you get far enough out, they don't produce any offspring. Um, these are two flies mating. They mate for about 60 seconds. And I'll tell you more about the mating here in a second. So is this because they're not mating or because of something that happens after mating? So that's pre-mating or post-mating incompatibilities. So another grad student, Emily, took this on, and this was another massive experiment. I feel guilty to some extent that I've had these people do so much work, but I've been involved in all of it too. So I'm showing here the inside of a female. And so here's mating happening, male on top, female below. Males actually insert and produce it inside the female, what's called a spermatophore. So this thing that looks sort of like a Bota bag, the black thing, that is a little uh, gelatinous substance or package that is males insert and sperm contains sperm. Sperm flow out of that package up into the spermathecae. Those are the female sperm storage organs where the sperm can stay for a month or more. Females then eject that spermatophore, and the sperm have to move over to the ventral receptacle, which is pictured here. And you can actually see if you look closely, they're actually single sperm coiled up inside each of those chambers. And then the eggs come down from the ovary and have to sit beneath that ventral receptacle for sperm to go into the egg, one sperm entirely into the egg, cause fertilization. And once fertilization happens, we can stain the egg and we can count whether or not they were fertilized. So basically, Emily, at each one of these steps, dissected flies to see where sperm had gone. Had they made it to the next step or not? And she continued on to look at whether the eggs were fertilized, whether they hatched, and then whether the sperm from males from other populations were successful or not as well. Whether there's if you put sperm in competition, which one wins? And 20 pairs of flies for every possible combination of these four populations that differ in how closely related they are. And I'll just show you a couple diagrams to try to illustrate what we were looking for. So if here's the fraction of flies that mate. So one would mean everybody mated. And this indicates how many sperm get passed in one mating. So if the mating is successful, they roughly pass 100 sperm um, because it's 10 to the log 2. And you can see all of these sort of within population crosses, they're pretty much the same. But there's a few that they mate, but they don't pass very many sperm. So those red dots are indicating cases where the sperm are not getting stored. So that's sort of like step one in the whole process. Next step would be, okay, they're stored, but they're not getting over to this fertilization site. That's the ventral receptacle. So again, there's a couple crosses where that didn't work so well. Whoop. So you can put all of that together and ask, where do these things break down? So this is a figure trying to illustrate from the most closely related populations to the most distantly related populations, which one of those factors is causing them not to produce offspring. And hopefully you can see the blue bars down here, that's mating. So mating, they happily mate. All these flies are mating. It's only when they're really distantly related they don't mate. What happens to the ones that are more closely related is that there's something to do with fertilization, sperm getting to the ventral receptacle. Some of the process of the sperm getting to the egg is interfered with, and it's asymmetrical. It's the two crosses, male versus female, aren't always the same. So what we conclude is that sexual isolation in these flies is happening uh, after the post-mating effects. It's not due to mating, it's due to these other factors. Um, so post-mating is evolving rapidly. It's not pre-mating. So it's not like they are prevented from mating, where, lots of things that use vocalizations, it's pre-mating that actually causes them to be isolated because they don't make the right call. Not these flies, they're happily mating, but their sperm aren't getting to the right place. And we think it's because the sperm and female storage organs are not matched. And I'll come back to that here in a second. Because there's one more factor, and that is that there could be hybrids and they're not fertile. That's post-zygotic isolation. And to explain how this connects to the drive, I have to give you a little cartoon to help you imagine what's going on, what we think is going on, what other people think is going on too. So us and most animals, 50-50 sex ratio is the norm. Lots of reason to think that's what you would expect to see. But if you get one of these um, selfish factors, so you can think of it selfish because it's getting itself into the next generation at a higher rate than it normally would. So it's these drive factors, X distorters, if they invade a population, you'll see more females and fewer males. So that will change the sex ratio. And as I showed earlier, that can lead to extinction. Of course, if it's extinct, we wouldn't see it, so we wouldn't study it. But it could also lead to polymorphism, which is stabilized in some way, which we think we're seeing. But there's an alternative, which I'll also show you seems to be happening, which is there could be other genetic factors elsewhere, particularly on autosomes or the Y chromosome, because they're losing in this sort of competition, that would suppress the drive on the X. And then you have what's often referred to as cryptic drive, because you wouldn't see it unless the suppressors were removed. Um, and that might lead to loss of drive, or it might lead to enhancers that would start the whole process over again. And so many people think this is an ongoing, constantly evolving arms race, drivers, suppressors, enhancers. And there are several examples now where we know this in some detail. We've learned something about these flies, but not as much as some other flies. But hopefully that's enough to make you wonder whether or not this hybrid sterility might be related to genomic conflict. Um, and that was an idea that people had now 35 years ago, that the drive suppressor co-evolving in isolated populations might result in hybrid sterility because when you put isolated populations together, they won't have the right drive suppressor combinations, and that will cause sterility. So if that was true, you would expect if you cross them, you would find cryptic drive. All of a sudden, you get drive where you didn't see it, and you would find some association between the genes or the genetic factors that cause sterility to also influence this sex ratio distortion. So we set out to test this idea because we knew from Sarah's work where she had looked at the crosses between, and then she'd also looked at the hybrids to see whether the hybrids were fertile or sterile. And what she found was what's known as Haldane's rule, which is that the females of these hybrids were generally fertile, the males were generally sterile, and the females became sterile if they were far enough apart, genetically more distant. The populations were genetically more distant. Um, so that gave us a sort of an angle to pursue that we could compare these populations which had hybrid male sterility. We had two possible combinations that are somewhat differently related. There's sort of this, these are the sites. Gambak is a field station in Peninsula Malaysia. Bukit Lawang is actually an orangutan rehabilitation site. Soraya is a field site that's hard to get to on the other side of the mountain range in Sumatra. So we collected flies from these sites and then looked for this association between sterility and sex ratio genetically. And that's a non-trivial task. Two different lab techs, most of who've gone on and gotten PhDs now, but they were involved in doing this. And because the hybrid males are sterile, you have to do a back cross in order to get fertile males to test the sex ratio and sterility. So that means these are diagrams of females here with all the bars of the same length. Blue is from the Bukit Wang, green is Gombak, and the two different length bars indicate a male having an X and a Y. So you mate the male to the female, then you mate the hybrid daughter to back cross to that one population, and then that son should have the genes on the X chromosome and all the chromosomes should have recombined. That's what the gray is indicating. And if there's recombination, we can then locate where things are in the genome. So we did this cross both directions. That's what GB versus BG. We switched the male, who is male, who is female. And we did the same with the Gomback Soraya, but in this case, we had to go another generation because the first generation backcross sons were sterile. So we had to go to the second generation backcross sons. A lot of work, a lot of flies, a lot of genotyping, a lot of work. But we found somewhat, I don't know, it came out better than I actually anticipated. That there was, and I failed to mention, these crosses, there were no flies that we started with that had distorted sex ratio. So there's no active drive among the flies we're using. Yet, when we look at the back cross sons, we see multiple different sex ratios of those sons. We see female biased sons, normal sex ratio sons, and males who are producing male biased sex ratios. In this first cross, all three phenotypes in both ways we did the cross. In the other cross, interestingly, I mean, this is what you would expect for Y drive, where you get predominantly male offspring. And on the other direction, we get predominantly female. I mean, again, it's 50-50 normal or female or normal or male. Then we located where in the genome are these things and found that in the closer cross, there's a major factor on the X chromosome that affects sterility. And at least one of those regions is also associated with distorted sex ratios. There's other factors on the autosomes, which we assume are essentially suppressors, that are also influencing the sex ratio. And there's one of these that's shared. The other cross, which is a little slightly more distant, you can see now there's more factors that are involved in sterility. So there's been more time, presumably, to allow that to occur. And yet there are two locations, the black one and the blue one, where there's association between sterility and the sex ratio effect. So none of these are shared across these sites, which implies to me that these are happening independently. They're evolving over time in these different sites. And yet they're associated with hybrid sterility and sex ratio distortion. So it's pretty clear that we can find cryptic drive in these flies that are otherwise not showing drive that are suppressed. And if you take the suppressors away, you see the drive. They because of the close cross, there's only an X-linked factor. That makes me conclude that it's happening first on the X, and then it's spreading to the autosomes over time. And I didn't talk about the sperm, but sperm are part of this, and it's the sperm length is changing, and if the sperm aren't the right size, they're not fertile. Um, and that it's all X chromosome is involved in all of this. So why? Where does this come from? So the last part of this is more looking at the genome of these flies and how does it compare to other flies. So there's a long history of people studying fly genomes, goes back over 100 years. T.H. Muller identified some very using banding patterns in chromosomes, salivary gland squashes, can identify similarities across different species of flies that he called molar elements. So they're different parts of chromosomes. And you can see in all of these different Drosophila, it sort of spans the phylogeny, I mean, many species of Drosophila, but this spans the major groups. They all have the same X chromosome as far as these elements go, with a couple exceptions where there's some fusions. Some of the arms of the chromosomes have gone and fused with the X chromosome. And it's interesting that those species, Subscura, Persimilis, Wilsoni, are known to have X chromosome drive. And that's been known for a long time. Lots of questions about it, but the fact that there's these new parts of an X have arisen suggests that maybe that could be some association between drive and novel X chromosome. So we wanted to know what's the X chromosome of these weird stalk-eyed flies. So Rick, my long-term collaborator now for many years, he's an American Museum still, and we came up with a technique, sort of pre-sequencing to figure this out using microarrays, and we were able to show that the X chromosome of our flies is largely the same as the left arm of the second chromosome in Drosophila. That's molar element B, this green bit. But there are also some parts of that chromosome that have either gone to autosomes or have moved onto the X. And most of these genes that have moved, we're able to show are expressed in the testes of the flies. And they include genes that are either duplicated of other genes or they're not found in other flies. So that's what an orphan gene means. So we wanted to, this is now the most recent trip, which is still a while ago, but most recent trip we made to Southeast Asia, we wanted to see whether there's more than one drive, active drive. I talked about the cryptic drive, which we uncovered, but now we want to see if we collect flies, can we find other groups that show drive? And so we caught, this is Kuala Lumpur, for those of you who know Peninsula Malaysia, that's what that metropolitan area is there at the bottom of this picture. These are collections from sites in the forests around Kuala Lumpur and then also from Brunei and Borneo. And the yellow is sort of normal sex ratio, red is extremely biased, and blue is this sort of mildly biased. And you can see in all places we find red, but these two, the Gombeck and Kanching, there's like 25% of the males we caught showed this strong drive. So we went out and we caught them, we brought them back, we scored their sex ratios, and then we created pools of testes or bodies of flies that we had scored the sex ratio. We took the RNA and we took the DNA and we sequenced it. And we're still doing it. But um, first thing we found was look at the testes of the males from Dolmani. That's the species we've done most of the work with. Either the extreme or the normal sex ratio males, pools of 10 males in each, pulling out the RNA, sequencing it, measuring gene expression, and we could find almost 600 genes that showed differences in expression, sort of substantial differences in expression. Those are almost all on the X chromosome, which may not be a surprise, but it's a large number of genes. I mean, that's more genes than you need to make testes. And there's other genes involved, but it's a lot of genes that are showing differential expression between otherwise males that pretty much are the same. A lot of those genes turn out to be either these orphan or duplicates. That's what we're calling novel. So they're sort of newly evolved. And we also found that many of them show evidence of moving onto the X or even onto the Y chromosome. And I'm not going to go through the details of how we were able to do that, but I'm just kind of summarizing, again, a fair amount of work. Most recently, we've assembled the genome this species and in particular the X chromosome for the drive male and the non-drive male. But this is first to show you a busy picture, I know, but the top is the standard fruit fly, Drosophila melanogaster, model fly for most fly genetics. And there's like, I think 800 genes are being visualized here, their order in the fly genome. This is our fly, the stalk-eyed fly, same 800 genes, and where they occur, either on C1 is one of the autosomes, C2 is the other autosome, and the X is the X chromosome. And you can see that this is confirming what we'd already knew, that the left arm and second chromosome is the X chromosome. As an aside, other people who are interested in sex chromosomes and flies have been able to show that the fourth chromosome in Drosophila is the ancestral X for all flies. And that seems to have just moved on to the middle of the other autosome. So it's not doing anything with our X. Uh, What's also being shown here in these bottom two figures is the distribution of transposable elements. And you might wonder, how do the genes move around? Well, we're pretty sure that those transposable elements are what are moving genes from one chromosome to another and also contributing to duplications of genes. But there's a lot. There's many more transposable elements in these flies than in your standard Drosophila. Now, if we just look at the X chromosome, is where things are interesting. So here we were able to sequence the X for both the drive and the non-drive and identify parts where those two sequences differ, and in particular where there were inversions. So flies are known for having inversions. So you take, you basically you read it right to left, and or you flip it around, you read it left to right. And so we were able to show that there are 6 differences, inverted regions between these two types of X chromosomes, which could be in the same fly. If a female's got both X's, she would have both of these. And they are very different. And there's a lot of ways I could describe the differences, but this second panel is showing the variation, sequence variation among individuals on the drive X or on the non-drive X. And the blue is the non-drive, the red is the drive. So across the drive, essentially, there's very little sequence variation. They're almost all the same. Whereas the non-drive, lots of sequence variation. The next panel is showing the difference or the divergence between the drive and the non-drive. So it's a huge amount of differences. This can only go up to one, this particular statistic. So you can see it goes up to 0.8. So it's large differences in regions of the X chromosome. And I highlighted this one panel because this is the region where there are some really extreme differences, but also there's five inversions that overlap in that region. And you might wonder, well, why would that be so many? I mean, if the factor that causes drive is in that region, then you might expect the inversions to accumulate. And indeed, we think we have a candidate for what's causing the drive. It's very hard for us to prove this, but I think the evidence is fairly compelling. And so this is showing across the X, these are genes that were differentially expressed, from the testes expression. That's DE RNA. The next one is there's differences in copy number. That is basically coverage that you can measure when you sequence DNA. And then one above that is genes that have both, the differentially expressed between drive and non-drive and differences in copy number. There's only 7 of these DEDC genes, and this is the only one that's in this region that falls within the overlapping inversions. And so we suspect that's likely critical. We know more about this. I don't want to go into a lot of detail about it because it will go beyond what I can explain to you. But we have pretty compelling evidence that it's a tandemly duplicated thing that's flipped in a couple of directions, and part of it is highly conserved, this sort of protein binding region, and it's associated with dosage compensation, which is associated with silencing X chromosomes. It's got a lot of factors associated with it that make it interesting as a candidate. And it's expressed in the testes. This particular region right here seems to be particularly important. So I'll just close with a couple more slides. I know I've kind of gone on here a little longer than I had hoped, but we are curious to know whether this active drive is present in other species. So Kim, who's still around, as I mentioned, teaching genetics, published this paper a few years ago, and we found that even though we thought every population was different, turns out there's multiple species that occur in both populations. Our sampling was not as sufficient. And so there's one species, so the species I've been talking about, we call species 1, got drive and non-drive, segregated, very different X chromosomes. This species, the drive and non-drive individuals show no difference that we can detect by sequencing parts of the X chromosome. And moreover, if we do this differential expression, we find essentially almost no differences in gene expression, and they're not on the X. So what we think is that this is a very recent case of drive, and at the moment, we actually have sent off flies to try to sequence this in detail to see if we can identify at the base pair level where there are differences. And we have pools of sequences, so we hope we can do this here in the next few months. So sorry if I've maybe taken a little longer than I'd hoped, but this last part, I think, in my mind, it makes it sort of interesting that the new X chromosome seems to be associated with drive. There's cryptic drive. There's more than one kind of drive currently, and the genetic basis may well be quite different, which is, I think, fairly interesting, which I set out to study these flies because I was interested in sexual selection. I had no idea there was going to be meiotic drive or to have something to do with speciation. This has been sort of a journey of discovery for me. And unfortunately, the guy who does the taxonomy passed away just like 2 weeks ago. So we don't have someone anymore who can put names on these species, which is a problem because there's a lot of species that need to be described. But these are the folks that did a lot of the work, and there's other folks involved as well as a sort of many, many undergrads who helped us take care of these flies. And if I see there's a chat and I blew past without stopping. Um, oh, yeah. I don't know if Jackie's still around there, but answering the question there, what do they feed on? If you're in Chiang Mai, you could very well see them because one of our populations came from Chiang Mai and they feed on rotting vegetation. But if you go through the forest, look for wild ginger. It's something that you can commonly find them on.
Okay. And what habitats are they generally found in other
We,
than near
oh,
water?
yeah, we find them typically near water. So there's some of the species that we've found this the sexually monomorphic ones actually we've caught along trails away from water. But otherwise, all the dimorphic species I think we've seen have been in riparian, right within 100 meters of a stream or other, body of water.
And approximately how large are they?
Body length is around 7 to 8 millimeters for most of these things.
Okay.
So, bigger than a housefly, but long and narrow. They look a little bit like an ant, sort of, if you just kind of see them, but they're also quite cryptic. I mean, you got to look hard because they'll be walking. They don't like to fly, perhaps not surprisingly, and they'll hover. So they'll jump up and they'll hover. Um, so yeah,
Okay. If I find any, I'll let you know.
okay,
All right. Well, this uh, Dr. Wilkinson, you can unshare now. We'll come back and we'll
sure.
open it up for Q&A. We already got started. Let me get a spotlight on you. Hold on. Here we go. All right, there he is. Thank you so much. That's a fascinating exploration where you go from. We're really getting down into the building blocks of what's going on.
Yeah, yeah. I
So
really,
Mike has if you have any questions, you can raise your hand and I'll call on you, or you can put it in the chat box. And we have a question from Mike. It says, flies and beetles are
yeah,
obviously very lineages. Have you studied the stalk-eyed beetles? This very odd anatomical characteristic is interesting convergent evolution.
yeah. I don't know that I've seen the stalk-eyed beetle. Do you know whether they're sexually dimorphic? Uh, I failed to point out that the Baltic amber, actually, the person who discovered there's enough Baltic amber specimens that you can show that they are sexually dimorphic. So you can find males and females and see the males have longer eye stalks. So I think that dimorphism is actually very ancient and they've lost it rather than gained it. But I don't know about the beetles. And I mean, other things have actually, elongations on their heads. Um, depends on what the what the sensory structures are out there. So why they might have done that. Whether it's associated with mating is what I'm getting at. There's some really great antlered flies in New Guinea that a colleague at Ball State studied that males have antlers, females do not. And similar sort of mating, I mean, they fight with the antlers just as you might think they would. And there's other flies that have other structures that are used for fighting.
All right. Do we have any questions up here from the peanut gallery? Somebody has their hand up. Who is it? Fred. Go ahead, Fred. You're muted, Fred. You're muted, Fred.
Now can you hear me?
Yep.
Okay, good evening. Um, I was just curious because when you showed the video of the adult emerging from the pupa and they expanded the eye stalks, so what's the how quick is that? That looked like it happened rather quickly,
Yeah, I think that was time lapse, so it's not as fast as that
right? That's why I
appeared,
asked.
but it takes, I would say, about a half an hour for their eye stalks to fully inflate, but then it takes several hours for the melanin, for that to darken and become solid. And in that span, they're very vulnerable. I mean, if you touch them, you'll mess up their heads. So, I mean, that happens. They usually eclose, I think, early in the morning, and then they crawl under something for that to occur, so they protect themselves.
I'm assuming they're pupating in the substrate somewhere on the ground, maybe.
Yeah, we've never found a pupae in the wild. We rear them in the lab on take whole ears of corn and use a commercial food processor to grind it up and autoclave it, and then we use that. They forage on that, and then they oviposit in it, and the larvae develop over a span of about 10 days, and then they'll crawl out and pupate in. We put them in little cups with cotton, so they pupate in the cotton, ideally, or we pick them out and put them in the cotton, and then they close about a week later. So,
Okay. How
and
many species did you guys experiment with when you were doing this?
boy, at various times, probably had at least 20 different species in the
Really?
lab.
Wow. Wow.
But some of them we would catch and bring them back, or people would send them to us, and quite a few just will not breed in captivity or not breed well enough to keep. So,
Hmm.
which is why, I mean, actually, I began trying to do this work with flies from Africa. I brought African flies back. I could not breed them and essentially was going to give up. But then I heard about these flies in Southeast Asia, decided to try those, and they much easier to breed. So, and as an aside, before I came to Maryland, I was actually in Colorado at Boulder, and I was initially going to try to work there, but I had to get a permit to bring the flies in, and because corn is one of the major commercial crops in Colorado, and there is a species in Africa that is a pest of corn. They said, no, you cannot bring them in. When I got to Maryland, I don't know who was at um, the permit place at the time, but they said, "Oh, no problem. I just want a couple samples for my collection." So it was quite a different. They could not survive out of my lab. In fact, we have a hard time keeping them going. They're very um, they're definitely not pests. None of the flies that we've ever brought in could be pests because they don't live on live plant material.
I see. Would it be safe to say that the other species that you have trouble trying to breed, would it be safe to say that the same mechanisms are at work with those species?
Yeah, that's a good question. It's really hard to say, particularly since as I tried to, went through all those different experiments we did, that we were able to show that there's cryptic drive. So that means that unless you do one of these crosses between populations, you would not know that that's happening. And so it's my suspicion is, given they all share the same X chromosome that we know, I would not be surprised that this is possible in basically all species, but you'd have to do crosses between them. And whether or not that happens very often, open question. We know in Southeast Asia, that soon to shelf region, so there's in Sumatra, there was a volcano that exploded, if I remember right, 120,000 years ago. Does that sound right? But Lake Toba, it's got a cinder cone in it. It's relatively recent, and it caused massive change in that part of the world. And so there undoubtedly were populations of all kinds of things, the rhinoceros, the Sumatran tigers, all those things would have been isolated to different degrees. And the water has gone up and down. So that used to be one big landmass. Now it's a bunch of islands. So it's the history there is a lot more dynamic than other parts of the world where you can find, certainly Africa, and where these flies occur.
Thank you.
Dr. Wilkinson, is there a maximum length of the eye stalks? I mean, you keep saying that the females were, they want to choose the largest eye stalks, but is there a point where the eye stalks were going to get too long and they're not going to be able to function?
Yeah, well, I mean, a couple of those pictures are about as long as anything we ever saw. And so definitely we have captured flies where the distance is twice the length of the body, and those flies do not fly very well. Um, so now I couldn't breed any of those really extreme ones, but in this experiment I did where I increased the stocks, I moved it about 20%. So I went from like 10 millimeters to 12 millimeters. So we could change it, and that was in 20 generations. Um, I don't know what the limits would be. I mean, undoubtedly it would have some negative consequences. But sexual selection is, you know, that is Darwin kind of had this knew this that what's good, what females are preferring or what works for males to fight doesn't necessarily help the males survive. I mean, they may well not survive as a consequence of that. So sexual selection and natural selection may work against each other. And we think in these flies, that's probably what's happening. And that probably limits the extreme case.
All right, thank you.
Mhm.
Okay, we have a question. Has anyone studied or speculated on the capability, parallax advantage, et cetera, that these types of eye structures might have?
Yeah, I don't know exactly what you're asking there, but I mean, that was some of the early ideas of why would you have eye stalks with overlapping field of vision. I mean, their field of vision has been mapped. There was a guy in Germany, Dietrich Burkhardt, who kept these flies, and that's how I knew I could go to Southeast Asia and catch flies that I could rear because he had been doing that. And he was a vision physiologist, so that's what he was interested in. And so early on, people suggested, well, maybe they're using it to catch prey, but they're not predatory. They have like sponge mouth parts. They just soak up bacteria and fungi from the leaf litter. They don't need eye stalks to do that. And it might help them avoid predation, respond to predators, see predators. But if that was the case, then I'm not sure you'd expect a big difference between males and females. And as I showed, in some cases, there isn't a big difference in males and females. So, um, you know, my suspicion, though, for why females have eye stalks is not necessarily because it's advantageous, but because they can't get rid of them. And I think it's because when they put their antennae out there, it's a one-way developmental street. Once you've done that, you can't go back. Because every other fly that has stalk eyes, females look like a normal fly. And it's only the males that have it, but they have their antennae in the middle of their heads. So I think that may be part of the explanation for why females, all of them have some amount of eye stalks. But our local Maryland stalk-eyed flies, they're very short-stalked, so they're not. I mean, if you caught one and looked at it, particularly in a microscope, you would know it has eye stalks, but it's not very impressive. I mean, and there's really no difference between males and females. They're pretty much the same.
All right. Questions, questions, questions? George, you look like you're getting ready to ask a question.
No questions, Your Honor.
Alan? Is Alan still on here? You got a question?
Thanks. I was interested in the function of the eyes, but the reason why Bronwyn's calling on me is, and this was sort of the most intense like focus in an area where I can't say I was relating so much. So now this makes this question even more significant. How the heck did you ever get interested in this in your life? Where did it start? And then how did it get down to this level of like detail and minutia? So, and I mean this kindly, but
Oh
I
yeah, no,
ask
no,
this of
no.
everyone who's presented.
No, that's fine. And maybe, well, as I think Brahman started, I was a, kid growing up in Northern California, and I was a bird watcher and, a naturalist. I mean, I liked to go out and catch things and look at them, and I was fascinated by any number of things. And I did a PhD on bats, and actually parallel work going on in bats while all this fly stuff is going on. But the flies started, I got a postdoc after I finished my PhD working on bats to go to England and study Drosophila, fruit flies, because I had 12 months and and I had an idea to test some models of sexual selection. So it was a very focused idea. But I discovered these stalk-eyed flies that they were brought to my attention, and talking to somebody. I was actually in Scotland at Edinburgh, a Drosophila person who just studied Drosophila song, because they sing by vibrating their wings, told me he'd been on holiday in Africa, in Kenya, and he'd seen stalk-eyed flies at this fish camp, and they were all over the place. So I thought, wow, maybe I should go look at the flies in Africa. So I just flew down to Africa. I mean, I got the cheapest flight I could get and went to where he said, and sure enough, there were the flies. Caught them, brought them back. This was sort of before there was no permit issues at that moment going to England. So we had them in the lab in Edinburgh, and people were really excited. Like, wow, these are crazy. But then, as I said, I then went from there to Colorado and could not get permission to bring him to Colorado. So I waited until I got to Maryland, brought him in. And it was at the beginning, I just wanted to ask questions about sexual selection. That's all I wanted. I thought it was going to be, 3 or 4 years, and then we'd be done and be moving on to something else. But I showed that we've discovered this sort of wild sex ratio changes that were, hard to ignore. And I was familiar enough with what had been done in Drosophila, other species, that I knew this was something that could be of interest. And so we just started, you know, going from one thing to the next thing to the next thing. And maybe one other explanation is in order to do research like this, it's not cheap. You've got to go places, you got to get the flies, bring them back. There's a lot of stuff to spend in the lab. You got to pay people salaries. So you got to persuade someone to give you some money to do this. And if none of this was funded by the National Institutes of Health, this was funded by the National Science Foundation, which is sort of basic science. So, and they do not fund renewals. They fund new projects. So you have to come up with a new project every time, and they're usually 3 years. So every 3 years, I came up with a new project, and that's part of the explanation for why. And I didn't tell you everything, but that's part of why there's, so much detail and maybe, asking somewhat different questions. Um, but yeah, and it's interesting. I mean, I did not start with any of this in mind. It was, but I don't have any regrets really. I mean, I think it was a lot of fun, a lot of interesting places to go, a lot of great people to work with too.
Just thought of a great, research, maybe, how scientists survive, the adaptations that scientists, do to adapt to get funding.
Yeah, I mean, ideally, you're not doing it, I never wanted to sort of either cure cancer or go to develop military applications. This was never part of what I had. I was, a naturalist. I just wanted to understand nature. And so maybe I got down a little too deep for some people, but that is still part of what is motivating for me.
Well, no, and
Thank you.
that's where we are now, especially with eDNA and everything like that, with the biology and going back even into the collections that we have and looking at those different relationships and taking a different look at it. I had a quick question also, is that when you had one female and the one male in the experimental setup,
hmm
does the female have any defense? Like if the male had the drive and she didn't want to mate with him, can she fight him or just say I don't want this?
Oh yeah. Yeah. No. Females have the ability to prevent males from mating, and even if they mate, they can also, if you recall, I tried to describe the males are passing this package of sperm into the females. Females can eject that right away. So they can control how many sperm they let males pass. And I didn't go through multiple mating sperm competition, but we don't have any evidence that females are choosing males in that regard. We do have evidence, though, that there's um, the drive, there's other things that are getting passed there that may interact with the sperm of a previous male and could affect the previous male's sperm ability to fertilize. So, but yeah, I mean, females can basically throw males off and prevent males from. They also can fly. I mean, they can just fly away. And um, in our little cages, that might be a little harder. They can't completely escape, leave them. But in the wild, they can just leave them, and males have no chance because it's they're not. If you go to the right place, you can see quite a few, but they're generally not abundant anywhere we ever went. Certainly in the daytime. It's only in the evening when you see these aggregations.
Gotcha. And I also appreciated that it was if I was following correctly, that the females have a lot of control in how the generations evolve, and they are fixing a problem, or I don't know if they were creating a problem by the selfishness of passing on, and then they have to fix it on the back end by selecting. They're kind of in control.
Let me see if I can ask him.
Yeah, go ahead.
Me, George?
Yeah, George, go ahead.
Oh yeah. You mentioned that you have a number of new species. I'm just wondering, do you have a substantial collection of each of these so that a taxonomist could go ahead and describe them? Are they dry mounted or in alcohol?
They're not dry mounted, so their majority are in alcohol or they're in the freezer. I mean, we basically bring a lot of things back and freeze them for using, but we have a lot of things in alcohol, and essentially we have all voucher specimens, I guess, for everything we ever captured. And it's on my to-do list when I retire, but that may not be realistic because I did learn. I mean, we started to try to describe a few of these and mounted the genitalia, but unless you're an expert and you're willing to get collections from other museums, I mean, it is a big chore, a task, and it's not part of my normal day-to-day work. So it really would be better for a specialist to do, but there are fewer and fewer of those specialists around. So it's not an easy thing to find someone who wants to do this. But they are a pretty charismatic animal.
I've always wanted to work on diopsids, I have to tell you, because they are cool to look at.
Yeah.
I work on chlorophids. They're not as exciting.
Okay. Well, yeah, I've got some specimens if you're interested.
Fred, did you have your hand up again? Go ahead, Fred.
I was just curious. You mentioned the specialist had passed away recently.
Mhm.
Was it someone from the Smithsonian? I just wondered.
No, he was from the Netherlands and spent a lot of time in Bhutan. Hans Fan is the guy. So he's yeah, he did his dissertation on African diapsids, and described. He actually worked on several Southeast Asian groups, but not this particular genus. But I corresponded with him. He told me my genitalia amounts were terrible, so I got discouraged. But yeah, I mean, it is something that you people who specialize in a particular group know kind of where the variation is. And I mean, we now, I think we have a good idea, but still, uh, the other problem is that, every collection, if they have access, has a diopsid and they have some name on it. And a lot of those names are wrong. I mean, I've seen in, I think it was in Oxford, I can't remember now where in Britain I saw, but the males were one species and the females were another species because they were so different.
Yeah,
So,
that happens a lot, unfortunately.
yeah,
And it really depends on all about funding, isn't it? Um,
yeah,
you
right.
know,
And time. I mean, it takes a lot of time to
yeah.
do this. And
Do we have anybody here in this country that specializes in these?
not that I know of, although I mean, I've been to the Natural History Museum and talked to, and I can't remember the names now of the of different people, but they're very, very good. I mean, they just have other things that they're
Yeah,
focused on. And it's not a major pest, so there's no economic incentive to really.
therefore, yeah, less time devoted to it, of course.
Yeah, and there's only, I mean, there are two species in North America, and the ranges kind of come together, and we're right near the junction of those ranges. But again, they're not common, but they're sort of, I think, kind of Mississippi east, maybe up to not much further north than Pennsylvania, actually. So we're close to the northern range, edge of the range.
Mhm. Well, as you know, there's so many other taxa across the entomological world where there's maybe one or two specialists in the world, that
Yeah,
know about that. So,
right.
um, yeah,
Yeah, and we did not get started on this to describe species, or that was never part of what we were trying to do, and that's why it's species one and species two.
mm, yeah,
But I mean, I certainly can describe some of them, but it will take some time, and I just need to find that time. Right now, don't have that time, but
right.
hopefully not too long.
Would you be willing to give us your contact information on the chat,
Yeah, sure.
an
Yeah.
email or something? Yeah.
Right. Yeah. Let me just I think I just copy this right here and I can um uh where's the chat? Right. There you go.
Yeah. Are you in the entomology department there by any chance?
No, I'm in biology, but
Biology. Okay.
um we're in the same college. I know everybody over there and it's yeah.
Well, that's
I know.
and then I have one last question for you.
Okay.
And you got to this from naturalist down into evolutionary biology, and then the graduate students that you're working with, and I was really excited to see all of the folks there. Were they coming to it? Are they more coming to it from the science side? Were they entomologists coming at it for this, or from their work, or did they become lovers of insects after this if they weren't already?
Um yeah, so I think it varies. Some of them, like the first guy I mentioned, Rick Baker, he was a grad student at Yale, and he was actually already then working at the American Museum because Yale had well, his advisor was on staff at the American Museum. And he went off and did postdoc. He did a couple postdocs actually in England, and he went in Berkeley. And then he managed to get a job back at the American Museum. So he is still at the American Museum At the moment, he's working on spiders, not on flies, but we're still involved. He's involved in this latest sequencing efforts that we're doing. And the two most recent postdocs, Josie and Kim, Josie did a PhD on flies. Kim did a PhD on pipefish. So very different, but um, I don't know. They got interested in the flies, somewhat different reasons, and they brought different skill sets, which was great because we were all together at the same time, which is very helpful. And the grad students, they were, I don't know that they had strong, well, one of them, Emily, she came from a lab at Syracuse that had worked under Drosophila, and so she knew about, she's the one that, dissected the inside of the flies to figure out how the sperm went. She was already, she'd done that with flies, with Drosophila flies. So she, as an undergraduate, so she came as flies. But now she works at UCLA as a development officer, so she's no longer doing science. So, I mean, people have come in and gone out in different areas, but some have, I mean, one guy who I didn't emphasize, didn't show a picture, John Swallow, he was a postdoc. He had done work on wheel running in mice for his PhD. Then he got working on these flies and got totally into the flies, developed a lab doing flies in Colorado, and became department chair. He was dean. Now I think he's stepped down from being dean. I think he's let his research program sort of decline, or he's not as active as he was. But yeah, I mean, there have been labs that have started up because we've given them flies. Because it's not that easy to go to Southeast Asia and go collect flies. I mean, it's a bit of a challenge to do that. And we still have some. We don't have anywhere near as what we used to have, but happy to share whenever anybody asks if there's a reasonable need or interest.
All right. Well, you never know where science is going to take you. When you start asking questions, more questions come up, and you follow those questions, and maybe you don't know where you're going to end up, which is the exciting part of the journey. And that's the same thing with the Natural History Society and Must Learn Thursdays. You never know where you're going to land. In January, remember, this is our last one for 2025. We start off the year with the snails of the Potomac. Snails of the Potomac. So we're going to learn all about that. Come back and see us. Thank you so much, Dr. Wilkinson, for sharing your knowledge with us. This was wonderful. And everyone, thank you for being here. Stay well, stay curious, stay dry, stay warm, and happy holidays.
Happy holidays.
Stay curious.
Yeah.
Stay curious. We'll see you. We'll see you next year.
Thank you very much.
You're welcome. Thank you.
Thanks.
Merry
Bye-bye.
Christmas.